Thank you very much for the generous introduction, and thank you very much to the awards committee for selecting me. It's really a great honor. And like Kyle said, um, look at the previous award winners, and it's really a who's who of people in our industry. So it's just really humbling to be considered in that same group. So thank you very much. Urban entomology and molecular ecology. These aren't two terms that you often hear together. And I think it's probably a shame that we don't often hear these two terms together, because these are two disciplines that I think have a lot to offer each other. Molecular ecology is a really big growing area that's taken over a lot of biology and has the potential to really provide incredible insights into the biology of organisms. And of course, urban entomology offers us many really amazing insects and other pests for study. But it's, you know, as an industry, as a scientific community, I think we've been a little slow to adopt the techniques of molecular ecology, but that's changing, and I think that's a good thing. So what is molecular ecology? Well, this is the Wikipedia definition. A field of evolutionary biology that uses a number of different approaches and techniques, including molecular population genetics. So if you look at this, let me get my pointer here. This word cloud, you see population genetics figures very prominently. Molecular phylogenetics, which is basically the relationships among organisms, and genomics has entered into it in a big way now. And uses these techniques to address traditional ecological questions. So ecology really looks at the interactions of organisms with each other and with the environment, and includes bigger questions about life history and behavior and so on. And so this has really made a big impact in many areas of biology. And in fact, there's a whole journal devoted just to molecular ecology, which has a pretty high impact factor if you're into those kinds of things. So there's tremendous potential for techniques of molecular ecology and approaches of molecular ecology to advance our understanding of the biology and management of urban pests. So Arnold Mallis, I really just know through his wonderful book. I'm sure that we all have on our bookshelves and refer to often. I certainly do. It's just a wealth of information about urban pests, integrated pest management. But even more importantly, probably, is just a wealth of information on the basic biology of a lot of our pests. And it's that biology that, you know, forms the basis for effective integrated pest management. So I don't know what Arnold Mallis would think of molecular ecology, but I'm sure he would appreciate the insights that we can gain from about the biology and management of different insect pests. So how can we apply molecular ecology to urban entomology? And I'll just touch on a few areas. These are areas that I've been active in, and there are lots of other areas. But one area is looking at genetic structure of populations. So this is basically looking at genetic diversity of populations. And so we've done work on bed bugs where we find very low genetic diversity within an infestation. And this tells us that infestations are started by very small numbers of individuals, probably a single female or the progeny of a single female in most cases. We contrast that with German cockroaches, where we find very high genetic diversity in infestations, because infestations tend to be started by much larger numbers of individuals. We can use these methods to look at dispersal and spread of populations. This is important to understand how infestations spread from one place to another. We heard a lot about insecticide resistance yesterday in student presentations. And so understanding how once insecticide resistance develops in an area, how it might spread to other areas is very important and can be addressed through these approaches. Invasion biology is a big area of molecular ecology, and most of our urban pests are invasive species. So retracing the routes of introduction can be important in understanding how species ended up in the areas where they've been introduced. And so we've done work on the eastern subterranean termite, which for us is not an invasive species, but it has been introduced in South America, and, Europe, and other places. And the Formosan subterranean termite, we've done work kind of retracing the roots of introduction of both of these species. And then looking at the invasion success of species. Why is it that some species are such good invaders that can be introduced to multiple places and become successful in those places? And so we've done work in that area, looking at some ants like Asian needle ant and tawny crazy ant. And we'll talk a little bit about the Asian needle ant later on. And then two other areas that I've spent a lot of time working on is looking at colony breeding systems of social insects. So social insects, of course, live in colonies that are headed by reproductives. And it is the workers in the colony that do all the work to support those reproductives. But the number of reproductives and the types of reproductives in colonies can vary between species and even within a species. And so using these approaches, we can gain insights into the breeding systems of these social insects, and talk more about that later. And then genetic fingerprinting of colonies. So in some species, it's difficult to say where one colony ends and another one begins, but we can use genetic markers to kind of apply a genetic fingerprint, allows us to separate individuals by colony. And some things that we can do with that is look at like the foraging areas of colonies. These are subterranean termite colonies. And we can look at colony densities, and we can track termite colonies over time in the field after an application of an insecticide to see what effect those insecticides might have on the entire colony. So for my talk, I'm going to concentrate on these two areas today. And first, I will just give you a little bit of background. So I think traditionally in these sorts of talks, people give some sort of biography and kind of how they got to where they are and who the influences were. And this is my biography slide. So I started off my scientific career doing research on the reproductive biology of fire ants at University of Georgia. And while I was there, I got to had the pleasure of meeting Ken Ross, who became a postdoc in our lab just as I was finishing, and he introduced me into a lot of the genetic methods. So I got my introduction to molecular ecology with Ken Ross. And there's an interesting story you can tell offline if you're interested. Ken is famous for discovering this social gene that separates the monogyne or single queen from the polygyne or multiple queen form of fire ants. And there's an interesting story about the role I played in the discovery of that. And then I spent a couple years in France working with Luc Passera on reproductive biology of Argentine ants, and then had a postdoc at University of Texas working on fire ants. And it was there that I started interacting with the Department of Agriculture and realizing that fire ants are not just really cool biological subjects, but they actually impact people's lives in a real way. And so I gained an appreciation for the impact that these species can have on people, especially in the urban environment. And that's sort of my introduction into urban entomology. And then I was hired, my first faculty position was at NC State, where I was hired as an urban entomologist to work on termites. And I had to think about, what am I going to work on when I get to NC State? You know, what can I contribute to study of termites? And I realized that there was a lot that we didn't know about the breeding systems and about colonies and things that I think were really ripe subjects for the application of the approaches of molecular ecology. So my sort of development as an urban entomologist and molecular ecology happens sort of simultaneously. So what I'm going to spend the rest of my time talking about is a little bit of work on subterranean termite colony breeding systems, the genetic identification of subterranean termite colonies, and some things we can do with that, looking at colony densities and colony level effects of termiticides, and then look at colony breeding structure in some invasive ants. So I've picked these things because I think some of them are just cool stories. I mean, I think they're really cool, so hopefully you will too. But I think some of them are actually really important for us to know as urban entomologists. Some of this is just very basic information about the biology of these species that as urban entomologists, we really should be familiar with. So I realized when I started working on termites, well, it doesn't take a genius to know that these are very cryptic species. They're hard to study. They live underground. You can have these foraging areas with chambers and nests that are interconnected through underground tunnels. So they're difficult to get at. So I was interested in learning more about the breeding structure of termites, and just kind of refresh your memory, or for those of you who don't think about termites all the time. This is the life cycle of typical subterranean termite, where you have male and female alates, that swarm, and then a male and female will pair up, and then they'll start a colony of a king and queen. That will be the sole parents of everybody in the colony. So the colony is initially a simple family with one mom and one dad and their offspring. And then if the king or queen dies, they can be replaced from within the colony by the progeny of the king and queen that develop into these secondary or neotenic reproductives. And there's usually many of these that inbreed within the colony. So colonies can be these simple families, the single king and queen, extended families with these multiple inbreeding neotenics. And we know from other work that occasionally you can have mixed families, and these can happen either through reproductives coming together when they found a colony, or more likely when established colonies fuse in the field. And so we knew all this when I started. We knew that there were these different types of breeding structures that occur in subterranean termites, but what we didn't really know was for any given population, what are the percentage of colonies that have simple families versus extended families? And how does that vary from one population to another? Again, these are cryptic species, so it's hard to go out and dig up the reproductives to see who's there. There's no way to really know where the nest is. These aren't like fire ants where you can go see the mound and dig it up and find the queens. But we can always find the workers. And so when we find the workers, we can genotype them, and then from their genotypes, we can infer the breeding structure and determine levels of inbreeding. So we've done a whole bunch of this kind of work. I'll just give you one example. One site that we studied in Charleston, South Carolina, where we had 3 species of reticulotermes, Flavipes, Virginicus, and Hageni, all co-occurring. And so we sampled colonies of all species in an area. We sampled everything we could get, and then genotyped them and determined family structure. You can see that the vast majority of colonies of all species were simple families. In Flavipes, about a quarter of the colonies were these extended families with inbreeding neotenics, and we even had some mixed families. And Hageni, a small percentage of extended families. Virginicus, they looked all to be simple families, but this is a more complicated situation. There's stuff going on behind here that I'll talk about more later. They just look like they're simple families, and it's more complicated than that. So that's just one population, and we've looked at lots and lots of populations. So one example of kinds of things we've done. So this is results from the eastern subterranean termite, Reticulotermes flavipes, where we genotyped populations from along the eastern seaboard, from South Carolina all the way up into Delaware, and then some other researchers had done some work in Massachusetts and Nebraska. And then you can see there's variation in the proportions of different types of family structures in these populations. And when you plot it out latitudinally, you see this nice gradient, where as you go from south to north, you have an increasing number of extended family colonies. That is, an increasing number of colonies with these inbreeding neotenics. So this tells us that there is this variation. It follows some sort of pattern here. It's there's a latitudinal gradient, but latitude is just a proxy for biological and environmental factors that also vary from south to north. So it tells us that these populations are likely responding to local ecological conditions in their areas. We have some idea of what those might be, but hopefully somebody will follow this up, maybe some students out there. I think it's a great system for understanding what are the ecological factors that shape breeding systems in these termites, which is a big question in termite biology and in social insects in general. When you have variation in the breeding system, what drives that variation? So I'm going to tell you about a type of breeding system that we discovered that was new and totally unexpected. And this is work I did with Kenji Matsura, a great friend and colleague of mine from Japan, who's an incredible field biologist. You can see he's quite at home here with a chainsaw, but he's also an incredibly deep thinker about termites and about biology in general. So this is work we started with a Japanese species, Reticulotermes spiratus. And this species forms colonies just like other subterranean termites, where you have the males and females fly from the nest, and then you have the primary reproductives pair up here. So we got a king and a queen. But what Kenji did, which I didn't do, which was actually go out and census the colonies, go find the reproductives in large established colonies. And what he found was quite surprising. So this shows the number of queens he found in colonies, and he always found multiple queens, and they were almost always secondary queens. He found one case where there was a primary queen together with secondary queens. And there was an average of 64 of these secondary queens. But when you look at the kings, when he did find a king, he found only a single king, and it was usually the primary king. So the original king that founded the colony. And so this is typical of what he would find is all of these large secondary reproductives together with the primary king, right? A single primary king. So we said that colonies can produce these secondary reproductives. And so we normally think of them as being the offspring of the original king and the original queen. So we would think that these are the daughters of the king that are inbreeding with the king. And in one case, we found almost 700 of these secondary queens in one colony together with a single primary king. So this guy was quite busy in his colony here. So again, we would normally think that these are all his daughters. But when we actually genotype them, we found something very surprising. So this is getting a little bit genetic-y, you know, but I think it's fairly simple, so just bear with me. But each of these lanes is the genotype of one individual at one microsatellite locus. And microsatellites are markers that come from the males and the females. They're inherited from both parents. So for example, the king here has two alleles. So he gets one allele from his mom and one from his dad. But we look at these secondary queens, these neotenic queens, we see that they only have a single band. Each individual has a single band. They're homozygous, right? And not only that, they're homozygous for alleles that are not shared in common with the king. So they are not the daughters of the king. The only way you can get this pattern is if these are the asexual progeny of the original queen. Basically, the queen is cloning herself. Technically, she's creating half clones. Each of these daughter queens is a half clone of the original queen that then mates with the king to produce workers that are sexually produced. So the worker genotypes here, they have one allele from the king in this middle range here, and then one allele from these queens, which all came from the original queen. And we looked at a number of colonies, and we find the same thing. Secondary queens are almost all produced asexually or parthenogenetically. Workers are produced sexually. Alates are produced sexually. So the system looks like this. Early on, you get a primary king and primary queen that get together, form a colony, the way that we normally think of subterranean termites doing, and then they mate and produce offspring sexually. At some point later on, the primary queen basically clones herself produces the secondary queens parthenogenetically. She stays together with them. The primary king mates with all of them to produce sexual offspring. Later on, the primary queen dies. The primary king hangs around. Secondary queens will produce other generations of secondary queens, also asexually. The king mates with them and produces sexual offspring. And you notice that the number of queens goes up in the colony. So this system is really cool because it allows the colony to grow faster and larger than it could if there was just a single queen present. The king is not mating with his daughters, as occurs in other subterranean termites. So there's no inbreeding that occurs. So any deleterious effects of inbreeding are avoided. And so we thought, and this is a really cool system. You know, it should be widespread in termites. Why don't other species use this system? And in fact, they do. So we looked at one of our local species, Reticular termes virginicus, that occurs all throughout the southern US. It's been studied forever. It's also of economic importance. It attacks structures. It has the exact same system where these secondary queens are produced asexually. And then a European group found another species of Reticular termes that has the same system, Reticular termes lucifugus in Italy. And then once this became known by other researchers, they began to look for it, and lo and behold, they began to find it. And this is a lot of work done by Yves Voisin, a Belgian researcher who works a lot in the tropics with higher termites, and he's found it in many species. So this is where we stand now with this asexual reproduction in termites. These are our 3 Reticular termes species. And then the higher termites, we have 2 species in this. So these are all the family Termitidae. We have 2 species in the subfamily Syntermitinae, 4 species in this termes group. And there's actually a group down in Japan of drywood termites, where there are populations where all the colonies are actually asexual, where there are no males in those colonies, that they're only females, and all the females are produced asexually. So asexual reproduction is widespread throughout termites, and this was totally unknown before and wouldn't have been possible to know without the use of these molecular markers. And we think it's much more widespread than we see here, just because people have not looked at many of these other species for this kind of system. So, you know, keep posted. I think it's going to be much more widespread than we see right now. Okay, so the next thing I want to talk about is genetic identification of subterranean termite colonies. So using these same genetic methods, we can genotype individuals from a colony. Because they are the progeny of a single king and queen, at least originally, they have a limited genotype. So they're genetically, they're genetically related, so genetically close together, but they're genetically different from other colonies, in the same habitat. So you don't have to know a lot about these markers to see that this group of 10 termites that were collected from a log are genetically different from this other group that was collected some 50 feet away. All right, so this is the way we can identify termites to colony and separate termites from different colonies. So we can use this to determine density of colonies. For example, around houses, each of these colored areas is a termite colony that occurred in the yard, or actually 2 of them in the house and in the natural area of the yard. And we can use it to track the fate of colonies after treatment to look at colony level effects. So this is a map that we're all familiar with, showing the subterranean termite pressure in different areas of the country. In the southeast, very heavy, in California, very heavy, and then in the middle, heavy to moderate, and then lighter as you go north. But this is very qualitative. We know that probably the mound densities are much higher in these areas where we have heavy density, but we don't have any good data to show that. So we set out to try to start collecting such data. And this is work done by my former student, Vince Parman, who many of you in the industry knew. And so Vince put out these these homemade monitoring devices, which is a piece of PVC pipe with some slots cut in the side and some wood stuff in the middle, and put out many around houses, and then sampled sampled them and then sampled out into wood debris out in the yard, and also any mud tubes that we saw, and then genotype them and determine the colony identity. And what we found in these 20 houses was that on average, there were about 9.5 colonies per property, and each property was about half an acre. So on average, there were about 25 colonies per acre, but there was a lot of variation between 10 colonies per acre up to 100 colonies. So this was a start, at least in this area of the country. You can tell homeowners, if you've got about half an acre, you can expect that you've got 9 to 10 colonies in your yard. It doesn't mean they're in your house. It's just in your yard. And then how does this compare to some natural areas? So we've looked at that. This was Schenck Forest in Raleigh, North Carolina, where we sampled over 3 years, and the different sort of shades of the dots here indicate the different years that they were sampled. And these were all the different places where we could find termites in this particular plot. And then once we do our genotyping, we can sort of fill in the gaps, connect the dots here and see where the colonies are. And you can see that the foraging areas in this plot were not very large, and that the colonies seem to respect the territories of other colonies, even over a 3-year period. But surprisingly, the number of colonies is quite high, about 170 per acre. And now, how does that compare to other areas? Well, for right now, we just have some work that's done by my former student, Mark Janowicki, who presented yesterday. He's a PhD student award winner. So congratulations, Mark. And this is a figure that he actually showed yesterday, but in a slightly different context. And so I'm just looking at the colony density here in his 2 plots, which were at Sam Houston State University Forest, which is in East Texas. It's 130 colonies per acre to 160 colonies per acre. So that's a pretty high density of colonies. And I think that's in Texas and in North Carolina, and probably everywhere in between has similar density. It's a lot of termite colonies out there. So the next thing I want to talk about that we can do with genetic fingerprinting of colonies is to look at colony level effects of baits. And we started this work with Centricon, which was fairly new at the time I began this work. And this is definitely the old system where you had the wood monitor, and then termites started feeding on that. You replaced that with the bait tube. And a common observation that pest management professionals had was that they put out the baits, they get termites showing up, they would bait them, termites would die or disappear, and then some months or years later, you get new termites coming back. And then the big question was, well, are these new termites that come back, are they from a different colony that's just moved into the territory that's vacated by the colony that died, or is this actually the original colony that's kind of recovering from treatment? And of course, we know that Dow said that this is a colony elimination system, so it has to be new colonies. And there, of course, there was some mark release recapture data to support that, but we weren't really sure on a larger scale if this would hold. And so I kind of went rogue. I did this without Dow's permission or knowledge, but with the help of a local pest management professional. So those of you who don't think about termites that much, think about a termite colony that's underground, it's feeding on multiple sources, including a house, and you apply a treatment here. And the idea with colony elimination is you apply some sort of a treatment where individuals will transport active ingredient back to the rest of the colony and cause that colony to be completely eliminated. So that's what we were looking for in this study. So this was an apartment complex that was under contract for termite treatments, and the company had Centricon baits. And so we followed it over 3 years. Each of these red dots indicates a station where we found termites and genotyped them, and each of the red dots is a different colony, except those that are encircled by different colors indicates a colony that occupied more than one bait station. So there were 23 colonies present this first year. Of course, they were all baited. And the next year we came back, and we were particularly interested to know whether stations that had been hit the year before and were hit again the second year, were those termites that were found the second year from the same colony that was present the year before, or from a different colony that had moved in. And except for this particular point up here, they were all new colonies that had come in. And this was the exception. In this case, this was a colony that had been given some bait, didn't eat very much of it the first year, and then the next year it had spread out into 3 stations. So it was given 3 bait tubes and it ate a lot more bait, and we didn't find that colony again. So in this particular study, we were able to track the fate of 36 baited colonies for at least a year. 35 of the colonies we never saw again after we identified them and they were baited. And 1 colony was found a year later, but not the following year. This is a huge number of colonies to be able to actually be able to identify individually and track over time. The previous work that had been done with mark release recapture, you know, on average, could follow 3 to 4 different colonies, and that was it per study. And those marks don't last very long. And they're not going to last 3 years. Whereas, you know, these marks with the molecular data will last as long as the colony lasts. It is its genetic fingerprint. So I think it just speaks to the power of these methods. So we've recently done some more work looking at area-wide treatments, and this is with the Valuron bait. And this is an area in College Station right behind my lab here, where we set out a grid of monitoring stakes, and then collected and genotyped everything. And then at the end of the 3rd year, we actually baited the colonies. And then we see a huge reduction in the number of colonies. And many of these colonies that were present on the 4th year were new colonies that had moved into the area and were just beginning to consume the bait. And those results are just shown here. We had a control plot where pre-treatment, you see, you know, there's a high level of, this is number of active stations. And then after treatment, the treatment plot, you can see that there's still a large number in the controls. Whereas with the treatment, you can see a large decrease in the number of active stations. And we're convinced that if you were to carry this out over time, you're going to get very low numbers over time. So you get sort of population-wide level control. So these techniques are important for not only showing us the effects on colonies, but they played a role in actually getting bait registration of, registration of a bait for as a pre-treatment in Florida. So it's a case where the application of these techniques has actually expanded the market of a bait. And it looks like Florida and probably EPA and maybe other states are reassessing some of their rules about registration of baits, and especially pre-treatment type applications. And these molecular methods may play a critical role in the future registration of some of these types of applications. And so may play an important role in actually bringing new products to market in the future. So we've also looked at the effects of non-repellent liquid termiticides on colonies. So there was some evidence that treatment with some of these non-repellent liquid termiticides could eliminate entire colonies. And so again, this was Vince Parman's work, where he set up these monitoring grids around houses that had termite infestations, and then went out and collected on a regular basis the termites, and then genotyped them to develop a map of all the colonies and where they were located around the house, including those that were actually infesting the house. And then did a treatment, and then followed, tracked the progress of in locations of these colonies for 2 years in the case of imidacloprid and 3 years in the case of fipronil. And the idea was if these treatments can eliminate colonies, then we shouldn't see the colonies again after treatment. They should disappear from the study and never be found again. And in fact, that's what we found in the case of fipronil, that all of the treated colonies, that these colonies that were actually infesting structures and were the targets of the treatments disappeared. We couldn't find any evidence of them again. Whereas the untreated colonies, those that were further away from the structure, we could find many of those again and find them on a repeated basis. With the imidacloprid, we also found colony elimination-like activity, although some colonies did seem to persist, at least over the 2-year period. And then we've done some more recent work with chlorinatriliprole, and this work was done in Texas, Ohio, and North Carolina. And we see, again, some evidence of colony level effects, colony treatment-like, colony elimination-like activity, but not quite as strong as what we've seen with fipronil and imidacloprid. Okay, now I'm going to switch gears completely and talk about ants. And start here with the Asian needle ant, Brachyponera chinensis. So this is a member of the Ponerine subfamily, which is an unusual subfamily, at least for urban pests and invasive ants. Most of our others are myrmecines, dolichotorines, and formicines. It's native to Asia, probably not this much of Asia, but a big part of Asia. And it was introduced to the US in the 1930s, but sort of went under the radar for a long time. And then I guess in the late 90s, early 2000s, it sort of kind of started to surge and became quite common in parts of South and North Carolina, and then spread from there. And it's now in 12 states. So this ant has some interesting characteristics. It's highly polygynous. I mean, there's lots of queens in colonies. It has a highly polydomous nest structure. I mean, there's lots of interconnected nests spread out through the landscape. It's got extremely dense populations that we can find where we find infestations in this park, Umstead State Park, where you can find these ants. Where you see them, the ground is like swarming with these ants. It looks like they just formed a super colony. And because of that, they have a strong ecological impact on native ant communities and other arthropods. We know that they really like termites, and they're termite specialists in its native range. And they eat a lot of termites in the introduced range as well. And unlike a lot of our urban pests, they do quite well in undisturbed forests as well as more urban habitats. So we were interested to determine if these colonies actually, the species form super colonies. And the way we did that was to have these transects where we walked that were 100 meters long, and then search all the wood debris for nests, which is where they tend to nest, and then collect the ants and genotype them. And we ran 4 transects in Raleigh, North Carolina, and then we ran a series of transects in 3 sites in Japan where they're native, including 2 on the big island of Honshu and 2 on Okinawa. So these are some of the sites we had in Japan, and there are transects in Raleigh. These are the different nests that we sampled, and then we can genetically fingerprint these. And this is what we found, is that colonies had a series of interconnected nests and could span a range of about 100 meters or so, but they weren't these giant super colonies like we see with Argentine ants or even Tony crazy ants. But each transect seemed to have multiple colonies. So that's in the invasive range. And then in the native range, we found a similar thing where colonies may span up to 75 linear meters, maybe it's a little bit smaller maybe than the invasive range, but again, they can be quite large and have many nests in them. So then we looked at the genetic structure of colonies, and I'm going to get a little genetic-y again. And if this is a little bit hard for you to follow, don't worry about it. I'm going to try to tie it all back in when I talk about the life cycle. But I want to give you some sense of how we actually infer the life cycle of this species. So first, in this panel here, we're looking at genetic diversity in populations. So we've got a number of microsatellite markers that we're using, and this is the number of alleles or different forms that are within those microsatellite markers. And this is at the population level. So these are the 3 Japanese populations, the 2 on the Big Island and the 1 on Okinawa. And you can see there's a little bit less genetic diversity in the introduced range in the US and in Okinawa, which is a small island, than on the Big Island in Honshu. But still, there's quite a bit of genetic diversity with between 7 and maybe 12 or so alleles per microsatellite locus. So we find something very different when we look at genetic diversity within colonies. So we're doing the same sort of analysis, but we're just looking at individuals within a colony. And the scale here is very different. So we're going from 2 to 6 here. And it turns out that there are only about 2 to 3 alleles per locus in each colony. So each colony has a very limited representation of the total genetic diversity within the population. And 2 to 3 alleles is the number of alleles you'd expect if each colony is started by a single queen who's mated to 1 male, right? But remember, these have multiple queens and they're spread out over large areas. And then finally, we can look at the genetic diversity within individuals. We do that by looking at heterozygosity. The expected heterozygosity is pretty high, but the observed is only half of that. So it's quite low. So one thing we were interested in is how related are queens to the males that they mate with? And we can determine this by genotyping the male, not the queens, and the sperm in the spermatheca that they have that contains the sperm from their mate. And this shows the relatedness between the queen and a mate. And so 0 would indicate the queens mate with unrelated individuals, and 0.5 would be that they mate with complete with siblings. And we see is that it's actually well above 0, and so they're mating with closely related relatives. And this leads to high inbreeding levels within the colonies. So this is a measure of inbreeding, which can go from 0 to 1, 0 being no inbreeding, 1 being completely inbred. And these numbers are quite high. And they're high in the native range as well as the introduced range. So here's where I'm going to tie it all back together. So from all that, we can infer that colonies are started by a single queen, mated probably to 1 male. And that colony is a single queen colony for a while. It's 1 queen. This is very typical of ants, this ant life cycle. But then at some point, the queen starts producing new queens and males that will mate within the colony. And then these queens will produce new queens and males that will also mate in the colony and spread out over the landscape, occupy areas up to 100 meters or so. And then at some point they will produce new queens and males that will fly away and start new colonies. So this is a very unusual breeding system for an ant. And it turns out that it's very similar to termites. Remember, termites are started by a king and queen that swarm, and then they can be replaced from within the colony by their inbreeding offspring. Again, these ants are termite specialists. They say you are what you eat. So maybe because they eat so many termites, they have sort of adopted a life cycle like these termites. All right, so this is the last species I'll talk about, is the dark rover ant, Brachymeryx patagonicus. It's native to a large area of South and Central America here. And it was first seen in Louisiana, I think in 1976, but it's rapidly spread throughout the Gulf Coast states and out west. It's in Arizona, New Mexico, California. It's even been found in Spain and indoors in other places in Europe. Around here, it's everywhere. It's incredibly abundant, and it's all around structures, and you'll see the densities are just phenomenal. So one thing we were interested in, so because this is a new species and it's a new urban pest, really, there's not much known about its biology. So we just started at the basics. We looked at 4 populations in Texas. We sampled from a number of different locations in each population. I just wanted to know these things form super colonies, or are these all genetically distinct entities? And what we found is that within each area, every place we sampled was a genetically distinct group. So this just shows the kind of genetic space occupied by individual colonies. So they are genetically separate groups. And I won't go into the details, but we can determine the number of queens within colonies just by genotyping workers, just like we do in termites. And we found that 90% of the colonies have just a single queen. Some have multiple queens, 2, maybe 3 on some occasions. And then we can determine the number of males that queens mate with. And about half the queens mate with a single male, and half mate with multiple males up to 4 males, which means that each colony is a group of closely related individuals that has basically one queen and maybe one male or two, maybe 2 or 3 males, but they're still very closely related groups. So we can apply our genetic fingerprinting abilities here and identify individual colonies in the landscape. And that's what we did around several houses. I'm just giving you 2 examples here. So these are all trails occupied by individual dark rover ant colonies. In this house here on the right, these gray areas are sort of foldouts of the side of the house to give you a better sense of the colonies that were trailing up the sides of these houses. These are all different colonies indicated by different colors, and you can go all around it. All 4 sides of this house, there were tons of ants trailing up it. And these trails were stable for at least a month. And so we did some behavioral tests to see if ants from different colonies were aggressive towards each other, and indeed they were. We just did some ant fights, put ants together and see if they fought, and rated them on a scale here of 1 to 5, with 1 to 2, just they're checking each other out. And then up here, they're fighting each other, spraying each other with formic acid. So they're not aggressive at all towards nestmates, but anyone else they don't like, and they fight quite aggressively. So these are relatively small colonies with a single queen for the most part. They form very dense populations, sometimes 1 colony about every 8 feet. And colonies are genetically distinct, and they're quite aggressive towards each other. And they do form these stable foraging trails, which does, I think, create some challenges for their control, which I'll talk about now. So we did some baiting studies to see if we could eliminate colonies through baits. So we could determine the number of colonies around structures, and then applied max force quantum ant bait at 4 to 6 locations around the house. So here's a house where we had 6 different bait stations located here with these red rectangles. And then we waited 28 days and saw which colonies were still around. So you can see in the control houses, we had a number of colonies, and in this particular one, there were some colonies that maybe moved on to some other things, but most of the colonies had very stable foraging trails, so they were still there. Whereas in the treatment colony, treatment house, and this is one example. You get lots of colonies, and then 28 days later, there was just this 1 colony left on the house, and it's right in between these different bait stations. Probably didn't feed on any of the baits. So these are the overall results of the controls. So pre-treatment and post-treatment, there's no change. But in the treatments, there was a 65% reduction in the number of colonies. And there was an 80% elimination of the original colonies present. But then in many of these houses, new colonies moved in to occupy the area that had been vacated by the colony that was eliminated. So we didn't get complete elimination, and I think in order to do that, you're going to have to probably put bait stations. This is a liquid bait. We had to put bait stations basically on every trail, and then come back often and see if there are new trails, and then bait those, because these trails do seem to be fairly stable. So wrapping things up here, I've talked little bit about breeding systems in termites and ants, what we can do with genetic fingerprinting of colonies, tracking colonies over time. And then I didn't really say much about invasion biology or spread and dispersal, but these are all areas that molecular ecology has a lot to contribute to urban entomology. There are many other areas as well, and there are a lot of genomic resources now available that provide very sophisticated means to get much more detailed information about the biology of these species. So I think a lot of this work will be making it into Mallis in subsequent editions. I know for a fact that some of it will be, because I'm revising the chapter on ants right now together with Robert Puckett. In fact, I'm overdue, so Stoy Hedges, if you're there, I'm really sorry. I'll get my chapter in soon. So I know that some of this work, some of the insights that we gained from molecular ecology are making it into the Mallis book, and so will be a good reference for others. And it's also encouraging to see a lot of students now and other labs, using techniques of molecular ecology. So I noted in our student presentations yesterday, there were 13 presentations, and 6 used to, at least to some degree, and in some cases, they were full-on molecular ecology studies, ranging from host meal identification to there was a phylogenetic study looking at insecticide resistance and microbiome characterization and so on. These are all applications of molecular ecological techniques. And so with that, I want to thank all the people who were involved in the work that I just talked about, and thank my lab, and thank you all for your attention. And I'd be happy to answer any questions.
I do have a couple of minutes for questions, so if anyone has any questions for Ed, I just butted in, but Kyle Jordan will be moderating them.
Yes, can we talk directly?
I recognize that voice.
You may.
Hey, Sam.
Yeah, don't see why not.
Hi, Ed, do you have any idea how long those males in those colonies last with the multiple queens, termite queens?
Oh, not really. So Kenji has some field studies. He's trying to get at that. So occasionally you can find secondary males. So they don't last forever, and the colony can outlive the the primary male, but probably not very long, because you don't find them very often, which suggests that when the colony gets to that age, it's probably not going to stick around very much longer.
Thank you.
But we're thinking it's probably on the order of 10 or more years.
Fairly impressive.
Yeah, and I think it's an interesting system. Why should males in that species live so much longer than the females, and what allows them to do that?
Whereas in ants, the queens live forever, and the males are only a flash in the pan.
That's right. Yeah, it's quite different.
Looks like there is a question in the chat. Is there a known correlation between termite family type and resource availability?
That's a great question. There's not anything known about that, although in our analysis, it looked like that areas where there was more wood debris, you tended to have more simple families than in areas where you had less wood debris. But that was a sort of statistical correlation. So it's an excellent question, and that's something that certainly should be followed up. But it's certainly possible that we don't have a good handle on that right now.
You're getting a lot of questions now. I don't know if you can see the chat. I'm going to try to keep up. Do you believe that the reasons for increasing genetic testing is the reduction in overall cost of doing the testing?
I'm sorry.
Do you believe the reasons for increasing genetic testing is the reduction in overall cost of doing such testing?
I think it's yeah, I think that's part of it, and part of it is just the technology is much easier, and people are doing more of it. So people in labs that didn't used to do this kind of stuff are doing it now. So it's a lot easier for more people to do it, and the costs certainly have come down.
And there's a question about the ages. So does this challenge the idea that the queen termite is the longest lived insect? Evidence from Georgia of 18 years, for example.
I don't think so, because there are certainly some higher termites, in those big mounds that live probably a very long time. Like Sanford was asking about the male termites, like Reticulotermes spiratus or Reticulotermes virginicus. They probably, I'm guessing maybe could live between 10 and 20 years. But I think we can see queens of other species that can live that long, and certainly queens of some of these higher termites can live a really long time. And kings too. So when we're talking about termites, we're talking about male kings and queens. So usually if the queen's going to live a long time, the king is going to live a long time too.
But we have some other questions that I think we can follow up on in email. We'll give you the contacts for those and the people who asked the questions. I think Ali would like to move on to the sponsor statements before we start the networking. So thank you again very much, Ed, and thank you to all the students who presented yesterday. Really appreciate it. And I'm going to turn it over to Ali.